This is me Arzu here and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 4 video of the alveolar bone, and in this video we will be discussing with the bone resorption, bone remodeling and the calcification. Okay, and in the previous video we have discussed with the ossifications, okay, intramembranous ossification and the endochondral ossification, and in this video we will be moving ahead in the chapter. Okay, so this is a part 4 video of the alveolar bone. So now without delaying much, let's start our today's video. Okay, so in the first topic, okay, so this is a calcification. Okay, so first topic is our calcification. Okay, and the calcification, it is the process of deposition of the insoluble salts in the tissue. Okay, so it is a deposition of the insoluble salts in the tissue. After that, mechanism of the calcification. Okay, so what is the mechanism of the calcification? Okay, so in this mechanism, so the first formed solid phase is the amorphous, okay, which finally change into the hydroxyapatite crystals. Okay, so first formed solid phase, okay, is amorphous, and this calcification happens upon the matrix. Okay, so as I have discussed in the previous video, okay, so first of all there is a formation of the matrix, and upon the matrix there is a calcification, okay, or the mineralization, okay, upon which there is a deposition of the insoluble salts. So first of all there is a formation of the solid phase amorphous. Okay, so upon that matrix, okay, there is a formation of the amorphous type of mineralization. Okay, so amorphous phase is there. Okay, solid phase is amorphous there. Okay, and finally that amorphous phase changes into the hydroxyapatite crystals phase. Okay, so here appears as a microscopic. Okay, so in amorphous phase it appears as a microscopic sphere 30 to 100 nanometer in diameter. Okay, comprising randomly packed. Okay, so in this amorphous, okay, as it changes into the hydroxyapatite crystal. So first of all in amorphous, when you see in the microscope, okay, so we can see 30 to 100 nanometer diameters, okay, sphere we can see in this amorphous stage. But when it changes to the hydroxyapatite, okay, so where there we can see the randomly packed apatite crystal, each of diameter 0 to 995, okay, 995, this is also nanometer, okay, nanometer diameter. Okay, so this is a hydroxyapatite crystals dimension, and this is the amorphous state dimension. Okay, so in the mechanism, just you should think that, okay, so amorphous form change to the hydroxyapatite form, means amorphous form change to crystal form. Okay, so now our another important point is a critical factor. Okay, so critical factor, it is a product of calcium and the phosphate. Okay, so inorganic calcium multiplied by inorganic phosphate gives the critical factor. So normally there is a insufficient concentration of the calcium and the phosphate ions in the blood and the tissue fluid to crystallize. Okay, so normally in the general state, okay, so this critical factor, okay, means the product of calcium and the phosphate is low. Okay, so there is a insufficient concentration there. Okay, but with the favorable condition, critical factor increases, and there is an increase with the increase of the critical factor. So increasing in the critical factor means there is an increasing of the calcium and the phosphate, so that leads to the deposition of the calcium, okay, or the calcification happens there. Okay, so in this critical factor, okay, so this can be framed into the MCQ question. So you just have to learn that, okay, so critical factor is just the product of calcium and the phosphate. Okay, so inorganic calcium and inorganic phosphate, and the concentration is insufficient. Okay, so this critical factor, that is a product of calcium and phosphate, so this critical factor is less, okay, in normally in the blood and the body tissue, in the tissue fluid, okay, to crystallize. But when the favorable condition comes, okay, at that time critical factor increases, and that leads to the calcification. So with the increase of the critical factor there is a calcification. Okay, okay, so now we have different theories of calcification. Okay, so first theory is a nucleation theory. Okay, another theory is a matrix vesicle theory. Okay, role of matrix vesicle, and another theory is a alkaline phosphatase theory. Okay, so we have particularly 3 different kinds of theories. Okay, and I have taken those points, okay, taken those notes from the Orbans. Okay, so I am following the Orbans here. Okay, so now we will discuss with the first theory, that is a nucleation theory. Okay, so simply in this nucleation theory you just understand that nucleation theory means formation of the nucleus. Okay, after the formation of the nucleus, and with the formation of the nucleus, okay, then after there is a mineralization, okay, so that act as a template, that acts as a center for the mineralization, okay. So we will be discussing properly here. So Newman in 1953 gave the nucleation theory, okay. So this theory, okay, so it is a theory of epitactic nucleation based on the concept of seeding or the epitaxy, okay. So this is a theory of epitactic nucleation, okay, that is based on the concept of seeding or epitaxy, okay. So don't go in detail in this point, okay, but you can just learn this, okay. So this is a theory of, and this is based on the concept of seeding and epitaxy, okay. You can just learn this much. After that, nucleus is formed probably in the relation to the collagen, effective in aggregating calcium and the phosphate ion, okay. So as I have told, okay, so this is very important point. So nucleus, okay, so nucleus is formed, okay, first of all, and that nucleus, okay, is effective in aggregating, okay, it is effective in aggregating calcium and the phosphate ion, okay. So that nucleus is capable, okay, it is capable enough to attract, okay, to aggregate the calcium and phosphate ion, so that there happens the mineralization, okay. So nucleus is a thing, okay, so it is a structure, okay, that is probably in the relation, okay, it is probably in the relation to the collagen, okay. So it is quite of related with the collagen, okay, and that act as a center, that act as a medium, okay, that is effective in the aggregating calcium and the phosphate ion, okay. So that is the main point of this nucleation theory, and this can be asked in MCQ. Newman gave this nucleation theory, okay. So new means you can learn this like, okay, so nucleus and new, okay, so they are quite similar when you pronounce them, okay. So in the first point, okay, so about in the ground substance, okay, so how the nucleus form in the ground substance, okay, so component like sulfated glycosamine glycans and proteoglycans, okay. So learn this: sulfated glycosamine glycans, sulfated GAGs and proteoglycans are responsible for the nucleation, okay, in the cartilaginous matrix prior to the mineralization, okay. So in the ground substance of the bone, okay, so in the ground substance of a bone we have sulfated glycosamine glycan and the proteoglycan, which act as a nucleus, okay. So they form the nucleus and they act as a medium center for aggregating the calcium and the phosphate ion, and finally the mineralization, okay. So that is the ground substance theory. And after that, in the collagen, okay, so in collagen, initial mineral deposit occurs at discrete sites in or on the collagen fibrils, okay. So first of all, okay, so in the collagen, okay, so there are the bundles of collagen. So first of all, mineral deposits, okay, so small minerals are deposited at the different points, okay. So if this is a collagen, okay, then we can see minerals are being deposited on the discrete side, okay. So mineral are deposited on different side, okay. There is no any kind of rhythmicity present there, okay. So at the discrete side, minerals are deposited in the collagen, and okay, so after that, dimension of the water filled pore space in the matrix of the collagen are crucial in allowing both ingress of the ion, formation of the ion cluster, and aggregation of the such cluster to form nuclei from which crystal growth can continue, okay. So in this collagen, okay, as in the ground substance you learn about the proteoglycans and the sulfated glycosamine glycan, but in this collagen you just have to learn that, okay, so the mineralization is discrete here, okay, so it is not regular here, it is discrete here in the collagen, and the thing, okay, that is a supplementing, that is helping in the formation of nucleus is that, okay, so there we can see the water filled pore spaces between the collagen, okay. So there is a pore spaces, okay, between the collagen bundles, and those pore spaces, okay, in the matrix of collagen, okay. So in the matrix of collagen we can see the pore like structure, water filled pore spaces are there, and that are crucial for the ingress of the ion, okay. So first of all, first point is the ingress of ion, means taking the ions inside, formation of the ion cluster, okay. So after taking the ions inside, formation of the ion cluster and aggregation of such cluster to form the nuclei, okay. So ingress of ion, formation of the ion cluster, and aggregation of the cluster to form the nuclei from which crystal growth can continue, okay. So that is the dimension, okay. So that is the way nucleus is formed in the collagen, okay. After that, mitochondria, okay. So mitochondria are earliest storage of calcium and phosphate. It is available extracellularly after cell destruction, supports the growth of the crystal. Okay, so in mitochondria, you just learn. Okay, so mitochondria are the storehouse. Okay, as it is a powerhouse, so it is also the storehouse of the calcium and the phosphate. Okay, so the content of calcium and the phosphate is more in the mitochondria, but when it is destructed, okay, by due to any kind of reason, okay, so let it be, so due to any kind of reason, if it is destructed, okay, so there is a release of those calcium and phosphate. Okay, and those calcium and phosphate, when comes extracellularly, they supports the growth of the crystals, and this supporting the growth of the crystal forms the nuclei and finally the mineralization. Okay, so we have studied about the ground substance, we have studied about the mitochondria, and we have studied about the collagen. Okay, okay, so now we will move to the another theory, that is the role of matrix vesicle theory. Okay, so how the mineralization takes place by the matrix vesicle. So in this first point, okay, so small membrane bound structure that is of 25 to 250 nanometer in diameter, lying free in the matrix. Okay, so matrix vesicle are actually small but membrane bound structure. Okay, so they are small but membrane bound structure. Okay, so they are like this. Okay, so they are spherical and membrane bound structure with a diameter of 25 to 250 nanometer. Okay, so this diameter, okay, so this diameter is 25 to 250 nanometer in diameter, and they are lying freely in the matrix. Okay, so if this is a matrix, so this is a matrix vesicle, if this is a matrix, okay, so in this matrix we can see the different matrix vesicle are present there. Okay, so another matrix vesicle is there. Okay, it contains its own content. So this is a ground substance. Okay, this is a sorry matrix, and in the matrix we can see the matrix vesicle. Okay, so now those matrix vesicles are the rounded outgrowth of the cell membrane of the osteoblast, chondrocyte, and the odontoblast, and this is very important. Okay, so they are actually the outgrowth. Okay, so if this is a, so just think this is a initial, that is odontoblast, chondrocyte, or odontoblast or osteoblast. Okay, so among those three, okay, so it is any cell, okay, among those. Okay, so now they are the, so now this matrix vesicle, okay, so this matrix vesicle are actually the outgrowths. Okay, so they are like this. Okay, so they are like this. Okay, so they are outgrowths. Okay, so they are outgrowth of this cell membrane, and when it is grown in full size, okay, so when it reaches to its full size, it just bud off. Okay, so it just cut off its connection with the cell and forms the matrix vesicle. So just think, they are the rounded outgrowth of the cell membrane of the osteoblast, chondrocyte, and the odontoblast, and this is very important. Okay, so remembering those cells will help you in the exam. Okay, after that, vesicles are rich in annexins, and this is also very important. So annexin is a chemical that forms the calcium channel, thus incorporating the ions within the matrix vesicle. Okay, so those matrix vesicle contains the annexin, and those annexins are the chemicals that are important, that are useful for the formation, okay, of the calcium channel in the matrix vesicle membrane. Okay, so those calcium channel would help in the incorporating the ion. Okay, so influx of the ion can be those from those channel. Okay, exchange of ion can be from those channel, so these are responsible for forming the calcium channels. Okay, in the matrix vesicle. So now matrix vesicle accumulate calcium, and their membrane furnish binding sites for the nucleation of the hydroxyapatite crystal. Okay, so this much in matrix vesicle. Okay, so they are the small membrane bound structure of 25 to 250 nanometer in diameter, and they are the outgrowths of the membrane of odontoblast, osteoblast, chondrocytes. Okay, and it also contains the chemical called annexin that is responsible for formation of the calcium channels on the membrane. Okay, and with those calcium channel membrane, okay, so it can ingress, it can influx the calcium ions, okay, inside it, accumulate the calcium ions, and also its membrane furnish the binding site. Okay, so there are also other chemicals that furnish the binding site, okay, on the membrane, and they form the nucleation. Okay, so here also we can see the nucleation. So all those theory are quite of interrelated with each other. Okay, so there is a internucleation, there is a cluster formation of the hydroxyapatite crystal, and that finally lead to the mineralization. After that, in a third theory, that is alkaline phosphatase theory. Okay, so it hydrolyzes broad range of the organic phosphate containing substrates and increase the local inorganic phosphate concentration. Okay, so this alkaline phosphatase is a enzyme. Okay, so it is an enzyme as it contains ASE. Okay, so ASE is there, so it is a enzyme, and it hydrolyzes the broad range of organic phosphate. Okay, so the main function of alkaline phosphate is the hydrolyzation. Okay, so it hydrolyze. Okay, just hydrolyze the organic phosphate, and those organic phosphate, okay, containing the substrate. Okay, so organic phosphate containing the substrate. Okay, and substrate can be anything, but just you think organic phosphates are hydrolyzed by this alkaline phosphate, and they increase the concentration of inorganic phosphate locally, and after that it resides in the matrix vesicle budding from the cell membrane of the chondrocyte, osteoblast and odontoblast. Okay, and this alkaline phosphatase, it resides in the matrix vesicle. Okay, and budding from the cell membrane of the chondrocyte. Okay, so same thing, it is for the matrix vesicle. I have told you here. Okay, so they are budded up, they connect, they disconnect their connections with the those cells. Okay, and they are formed from the cell membrane of those chondrocyte, odontoblast and osteoblast. Okay, and active markers of tissue mineralization. Okay, so this alkaline phosphatase is a active marker for tissue mineralization, and this is very important question for your MCQ exam. Okay, after that, okay, so we have discussed with the calcification, we have discussed with the theories of calcification. First was nucleation theory, another was matrix vesicle theory, and third is alkaline phosphatase theory. The function of alkaline phosphatase, it just hydrolyze the organic phosphates, okay, containing some substrates, okay, some organic substrate, and that increases the local phosphate concentration, and that forms the nucleus. Okay, finally the mineralization happens there, and it resides in the matrix vesicle. Okay, which is budded up from the cell membrane of those cells, and it is active marker of the tissue mineralization. Okay, so now there are also inhibitors for the calcification. Okay, so you may think, okay, why the soft tissue? Okay, soft tissue do also contain the collagen, and also the bone contain the collagen. Okay, but why the soft tissue collagen do not calcify? Okay, so the reason is due to the presence of the inhibitors at the soft tissue collagen. Okay, so it is because of the inhibitors. Okay, and they are all okay. So one reason is the inhibitor, and another reason is, so they are tightly packed. Okay, that don't allow the calcium to come in interfibrillar space, and no calcification. Okay, so you may just confuse. Okay, so the collagen, okay, so as the bone is stronger, as the bone is harder, okay, so there must be the collagen more tightly packed. Okay, but it is not so. So as it is written here, the collagen are more tightly packed in a soft tissue. Okay, so that cause the, so that just close the entry point. Okay, so it just close the entry of the calcium inside it. Okay, to come inside it. Okay, in the interfibrillar space, and no calcification happens there. Okay, so they are the two reasons for the inhibition of calcium and calcification in the soft tissue collagen. So the now inhibitors, okay, so chemicals are there. So one is citrate, another is magnesium, albumin. Okay, so it is blood protein. Okay, and pyrophosphate, bisphosphonates and proteoglycans. So you can learn this. Okay, so they can ask you in the MCQ. Okay, so tick the inhibitors of the calcification, so in any of the option. Okay, so they can put those chemical name. Okay, okay, so now we have completed with this calcification. So now we will start our another topic of today's video, that is bone resorption. Okay, so and all those topics are quite of general topic. Okay, so it is not a compulsion to study in this oral histology, but they are actually the general topic you must have studied in the general basic science subject. Okay, in anatomy, histology, okay, or physiology. I think you have studied this. Okay, so they are quite of general topics. Okay, but also as it is given in Orbans. Okay, and we follow Orbans, okay, properly, so we are discussing it in a detail here. So now we will move to the bone resorption. Okay, so now bone resorption, so simply bone resorption means destruction. Okay, resorption, destruction of the bone. So it is a removal of the mineral and the organic components of the extracellular matrix of the bone by the osteoclast. Okay, so osteoclast is a bone resorpting cell. Okay, so not only mineral but also the matrix. Okay, so that is important here. After that, phases of bone resorption. So we have phase 1, phase 2 and phase 3. So in the first phase there is a formation of the osteoclast precursor. In second phase there is activation of the osteoclast, and in the third phase there is resorption by the activated osteoclast. Okay, so they are the three phases of the bone resorption. So first of all there is a formation of the precursor cells, okay, of the osteoclast. Okay, so osteoclast precursor cells are formed. After that those osteoclast precursor cell, okay, forms the osteoclast, and those osteoclast get activated, and finally those activated osteoclast, okay, resorb the bone. So that are the three phases of the bone resorption. Okay, after that sequence of the bone resorption. So first sequence, okay, so in the first step, so alteration in the osteoclast. Okay, so first of all there is alteration in the osteoclast. Second, there is a removal of the hydroxyapatite. Okay, second is the removal of hydroxyapatite. Third is a degradation of the organic matrix, and fourth is a removal of the degradation product from the lacunae. Okay, so we have four different steps of the bone resorption. So first of all there is alteration in the osteoclast. After that there is removal of the hydroxyapatite crystals, and with the removal, okay, so there is a degradation. Okay, after that there is a, when you will just remove the hydroxyapatite, there will be left matrix. Okay, organic matrix will be left there. So degradation of organic matrix, and after that removal of the degradation product from the lacunae. Okay, so the thing you have degraded, you have destroyed. Okay, so you should have to remove that. Okay, so that is the proper four steps of the bone resorption. Okay, and all those four steps are included in this chart. Okay, this flow chart, and this flow chart is very very important chart. Okay, if you want to learn this bone resorption, okay, so in exams you just draw this flow chart, and this is a flow chart of Orbans. Okay, I have copied this flow chart from the Orbans, and there I saw, okay, so it was clearly explained everything. Okay, so you shouldn't have to study the text in the book, but you just follow this flowchart and remember this flowchart properly, so you can make same flowchart in your exam and get the proper marks. So now let's study the flowchart. Okay, so in the first point, okay, so there is a formation of the osteoclast progenitor in the hematopoietic tissue. Okay, so as in the first phase, okay, so in the first step, as it is alteration of the osteoclast. Okay, so before the formation of the osteoclast, you need to there should be presence of the osteoclast progenitor cell, precursor cell. Okay, so this is the first step of the bone resorption. So formation of the osteoclast progenitor in the hematopoietic tissue, vascular dissemination takes place there. Okay, and generation of the resting pre-osteoclast and the osteoclast in the bone, and activation of the osteoclast. Okay, and this activation is due to the interaction of rank-L and the rank. Okay, so don't go in depth, but just remember, due to the interaction of rank-L and the rank. Okay, so they are the receptor present on the cells. Okay, so one is a chemical, another is a receptor. Okay, so they just interact with each other and they activate the osteoclast. Okay, so just in the first step we learned that, okay, so osteoclast progenitor cell is formed in the hematopoietic tissue, and due to the vascular dissemination. Okay, so when there is a proper vascular supply, okay, so it just differentiate, okay, into the pre-osteoclast and the osteoclast. Okay, and after that, after degeneration, after generation, okay, so after generation of this pre-osteoclast and osteoclast, after that there is a activation. Okay, so that is the first step, okay, of this bone resorption. After that, the migration of osteoclast to the resorption side. Okay, and migration of the osteoclast to the resorption side, and another chemical MMP9 helps in migration. Rank L is to OPG, okay, reaction. Rank L increases the osteoclast formation. Okay, so here you just learn that. Okay, so in the next step, okay, so there is a, after the activation of the osteoclast, it is migrated to the resorption site. Okay, and that is helped by the MMP9. Okay, so learn this much. MMP9 is the chemical that help in the migration of the osteoclast, activated osteoclast, to the resorption site. And this rank L, okay, so this rank L increases the osteoclast formation. Okay, so more osteoclast is formed by this rank L. After that, in the next point, okay, so in the B point, okay, so the formation of the ruffle border and the sealing zone. Okay, so there are two structure, one is ruffle border, another is sealing zone is formed. Okay, after the first step, this ruffle border, so osteoclast, as I have shown in the previous video. Okay, so I will also show here. So in this point, okay, so first of all there is a formation of the ruffle border, okay, so there is formation of the ruffle border, and after the formation of the ruffle border and the sealing zone, in the next step there is attachment of the plasma membrane of the osteoclast to the bone matrix through the integrins alpha, okay, and the name of the integrins, and it is not quite important, you should not have to learn much, okay, this is in quite detail, you just remember integrins, okay. After that mmp13, that is secreted from the osteoblast, okay, so that is degrading the osteoid and exposed site for the osteoclast attachment, okay. So now I will explain this point properly, okay. So in this point, okay, so there is a formation of the two structure in the osteoclast, okay. So in the osteoclast we have discussed, okay, so there is one smooth side, okay, and at another side there is a ruffle border we can see in the osteoclast. So in the osteoclast video, in the bone cell video, we have discussed with this, okay. So there is a formation of the ruffle border and the sealing zone at the site of the resorption, okay. So I'll show you I'll draw a schematic diagram here, but first of all listen to me, okay. So in this osteoclast, okay, so I'll draw a small picture here, okay. So just think this is a osteoclast, okay, and this is a ruffle border here, okay. So now in this ruffle border, okay, we can see the sealing zone here, okay. So at this side, okay, we can see the sealing zone, and this is a resorption side, okay. So this is a resorption side, this is a resorption side, this is a bone, okay. So this structure is a bone, and at this portion, okay, so there is a resorption, okay. So there is going to be resorption, so there is a formation of the ruffle border. So this is a ruffle border, and sealing zone is just adjacent to that ruffle border, okay. Sealing zone function is to just seal the osteoclast, okay, at that site, okay, and the ruffle border, it increases the surface area, okay, for the exchange of the chemicals, okay, for the resorption. So this ruffle border and sealing zone, okay, so the attachment of the plasma membrane osteoclast is through the integrins, this is important here. And mmp13, okay, mmp13, it is a chemical which is secreted from the osteoblast, it is degrading the osteoid, okay. So osteoid that is present in the bone, okay, in this exposed portion of the bone which is going to resorb, okay. So at that portion mmp13, it just degrade the osteoid and facilitate the osteoclast attachment, okay. So in this second point, just remember ruffle border is formed, sealing zone is formed, attachment of the plasma membrane of the osteoclast to the bone matrix, and with the help of integrins, and mmp13 is responsible for degrading the osteoid and facilitating the osteoclast attachment, okay. So that is only here. After that, in another step, okay, so osteoclast synthesizes, okay. So now osteoclast synthesizes some chemical, so those chemical, so cytoplasmic carbonic anhydrase, okay, cytoplasmic carbonic anhydrase 2, okay. So and about this enzyme, okay, I think you have learned, okay. So this carbonic anhydrase, this enzyme forms the H plus ion, okay. So H plus ion, okay, so when there is a release of protons, and those protons are released in ruffle border and low pH, okay. So those protons, those H plus ion, they form the environment quite acidic, means low pH is there, okay, and dissolution of the hydroxyapatite takes place, okay. So this step is quite easy. So osteoclast synthesizes cytoplasmic anhydrase 2, and this cytoplasmic anhydrase 2, okay, this, I think about this enzyme you have been studied properly in the pharmacology also, I think, okay. So I will not discuss much about this enzyme, but I can say just this much, this is an enzyme which is responsible for making that environment quite acidic by formation of the protons, okay. So by the formation of H plus ion at that side, it makes the environment quite acidic, and those acidic environment is responsible for dissolution of the hydroxyapatite crystals, okay. So now I will move to the another point. So now also other enzymes like cathepsin K and mmp9 synthesized in a rough endoplasmic reticulum, transported to the Golgi complex, moved to the ruffle border in the vesicle, and contains released into the extracellular lysosomes, okay. So degradation of organic matrix. So it is quite it is written in quite longer form, and I think you have learned, okay. So how protein are secreted, okay. So this is a process of translation, okay, secretion of the, not translation but the secretion of the protein, okay. So enzyme cathepsin K and mmp9 are also equally important in this bone resorption, you just remember that much. After that, removal of the degradation product, free organic and inorganic particle endocytosed from the lacunae across the ruffle border into the osteoclast. Okay, so the remove, so those degradation product, okay, that are formed, okay, and that contain organic and inorganic particles, okay, and those are endocytosed from the lacunae. Okay, so it is endocytosed from the lacunae across the ruffle border. Okay, so around the ruffle border, across the ruffle border, it is just endocytosed into the osteoclast itself. Okay, and they are packed in a membrane bound vesicles released by the exocytosis. So the content, okay, which is formed here, okay, so they are finally again coming inside the osteoclast, okay, by this from this ruffle border, okay, from lacunae across the ruffle border into the osteoclast. Okay, so just remember those degraded product again comes into the osteoclast, okay, and they are packed into the membrane bound vesicles and released by the exocytosis. And trap is an enzyme, okay, we will be discussing with this trap also helps in the migration of the osteoclast to the adjacent site. Okay, so this trap have more important, okay, in this exocytosis of those degraded product. And finally there is apoptosis of the osteoclast. Okay, so finally the osteoclast it just apoptoted. Okay, so it is died. So now I will just say once summary of all those steps. Okay, so first of all, there is a formation of the osteoclast progenitor cell in the hematopoietic tissue. With the vascular dissemination there is a generation of the pre-osteoclast and the osteoclast. Okay, and they finally activate the osteoclast. Okay, and the activation of the osteoclast is by the rank L and the rank interaction. Okay, so interaction of rank K, rank L and rank. Okay, so that interaction activate the osteoclast, and with the activation of the osteoclast they are finally migrated to the resorpting site, and the migration is helped by the MMP9, that you should remember. After that, in the another point we have studied that when the osteoclast is reached, okay, so when it is arrived at that resorpting position, okay, resorpting site of the bone, so there it forms the two structure. Okay, one it forms the ruffle border, another it forms the sealing zone inside it. Okay, and those two structures are responsible for the attachment. Okay, and the sealing zone is quite important for the sealing of the osteoclast to the bone, and this ruffle border is increasing the surface area for exchange of the material. Okay, for the secretion of material in that space. And this attachment of the plasma membrane to the osteoclast is by the integrins there. And after that, MMP13, okay, which is also secreted by the osteoblast. Okay, it just degrades the osteoid, and it at the and also it facilitates for the an exposed site for the osteoclast, also facilitate for the osteoclast attachment. Okay, so just remember this much. Okay, so it is quite I'm just teaching this in more detail, so it may feel quite difficult to understand, but it is very easy. Okay, so integrins for attachment, MMP13 for degradation of osteoid. After that, osteoblast synthesizes different cytoplasmic carbonic anhydrase, and those are responsible for secretion of proteins at that site that makes the environment acidic, and the acidic environment degrades the hydroxyapatite crystals, dissolution of hydroxyapatite crystals. Okay, so dissolution of inorganic content there. After that, enzyme cathepsin and MMP9. Okay, so cathepsin K, especially cathepsin K and MMP9 are synthesized from RER, transferred to the Golgi complex, and they are quite finally secreted from the cells, and they are also responsible for the dissolution of the organic. Okay, so this acid dissolutes the dissolution, so this acid dissolutes, okay, the inorganic content, but this enzyme they just degrade the organic content. Okay, so that is the difference. So enzyme degrades the organic, and this H plus ions, okay, they degrade the hydroxyapatite. Okay, and after that finally there is a degradation product formed, and that degradation product are taken inside again into the osteoclast, and they are packed into the membrane bound vesicles, and they are finally exocytosed. Okay, and trap is helping in that exocytosis, and finally there is a apoptosis of the osteoclast. Okay, so now we have completed this bone resorption. Okay, so once you can look this chart and you will be more clear. Okay, so now we'll study about the trap. Okay, so tartrate resistant acid phosphatase is the full form of this trap. Now this in this trap, okay, so trap full form is tartrate resistant acid phosphatase. Okay, so they are latent inactive proenzyme. Okay, so they are latent inactive proenzyme cleaved by the enzyme cysteine proteinases. Okay, so this is also important. So this is a enzyme cysteine proteinases which are responsible for activation of this trap. Okay, so they are quite of inactive proenzyme. After that, this trap have two function, extracellular and intracellular function. Okay, and about this trap, okay, so they can ask you in the short note. Mostly they are asked in a 1 marks or 2 marks question. Just write a short note on this trap, so you can write like this way. Okay, so I have written in detail here, but you can make in your pattern. Okay, in exam. Okay, you can just sort it and write. Okay, so there is extracellular role of the trap and intracellular role of the trap. So in the extracellular role of the trap, accumulate extracellularly adjacent to the ruffle border. Okay, after that, osteopontin. Okay, so osteopontin, bone shallow protein and osteonectin act as a substrate for the trap. Okay, so they are the substrate for the trap. Okay, so it is an enzyme, so we have substrate and the product. Okay, and this osteopontin, BSP, osteonectin act as a substrate for the trap. After that, osteopontin, it enables the osteoclast to adhere to the bone surface by binding with the integrins. Okay, integrins is B beta 3. Okay, don't remember this, just you remember integrins. Trap remove phosphate group from it and disrupt the adhesion of the osteoclast to the bone. Okay, and it regulates the osteoclast adhesion. Okay, so here you should learn that. Okay, so osteopontin, BSP and osteonectin, they are the substrate for the trap, and this osteopontin is enabling the osteoclast to adhere to the bone surface. And as I have told, attachment is quite facilitated by the integrins. Okay, so integrin is attaching that, and it is helping by the osteopontin. Okay, it is helped by the osteopontin. Now this trap is also present adjacent to the ruffle border, and this trap remove the phosphate group. Okay, so it remove the phosphate group from the osteopontin, and this removing the phosphate group from the osteopontin, the attachment, okay, is also disrupt. Okay, so it is also disrupt. So adhesion of the osteoclast is also disrupt at this position. Okay, so finally it is helping in the uh, is helping in the disruption of the adhesion of the osteoclast to the bone. Okay, so finally it regulates the osteoclast adhesion. Okay, sometimes there may happen the more adhesion of the osteoclast at the bone. Okay, so it can resolve more surface area of the bone. So to prevent that, this trap is helping here. Okay, so it regulates the osteoclast adhesion. Okay, so in this point, trap can hydrolyze the bone matrix and release the pyrophosphate. Okay, also it releases the pyrophosphate. Okay, one way is this. Okay, by removing the phosphate group from the osteopontin, it just disrupt the attachment of the osteoclast. Okay, so it is decreasing the resorption, okay, of this osteoclast of the bone, okay, by the osteoclast, and also it decreases the resorption. Okay, inhibits the resorption by this pyrophosphate release. Okay, and this is released from the bone matrix hydrolyzation. Okay, and which is done by the trap. Okay, so this is the extracellular role of the trap, and the intracellular role of the trap is trap containing vesicles. Okay, it fuses with the transcytotic vesicle, transporting the matrix degradation product from the ruffle border to the FSD, functional secretory domain, okay, of the osteoclast. Okay, so as I have told, okay, so this is also responsible for the exocytosis. Okay, so it is helping in the exocytosis. So this trap containing vesicle, it fuses with the transcytotic vesicle. Okay, and they transport the matrix degradation product at the FSD. Okay, from the ruffle border to the FSD. Now dispose the product of the bone resorption in the transcytotic vesicle, and once the trap is exposed to the physiological influences present in the body fluid, binds to the 2 macroglobulin and the structure of it comprised, leading to the inactivation. Okay, so this is a fate. Okay, this is not under the intracellular role of the trap. In the intracellular role, you just remember it is helping in the exocytosis. Okay, and this is a proper pattern. Okay, you can just so I have written in the proper way. Okay, so when you just read this note, you get the proper overview what is happening there. Okay, but for remembering, you can just remember it is helping in the exocytosis. Okay, so the proper way is this transcytotic vesicle. Okay, transporting fuse with the transcytotic vesicle. Okay, and transport the matrix degradation product from the ruffle border to the FSD. Okay, so that is the proper way. You can learn in your way. Okay, so I have written here. Okay, so now our another topic of okay, so our last topic of this video is a bone remodeling. Okay, so in the this bone remodeling, okay, so bone remodeling is the coupling process. Okay, so coupling of the formation and the resorption. Okay, so there is also the bone remodeling simply means, okay, so there is also formation of the bone and also there is a degradation of bone, and both are going simultaneously. Okay, so that is the remodeling process. Okay, so it prevents the accumulation of the damaged and the fatigued bone by the regenerating new bone. Okay, so that is one function. Allow the bone to respond to the changes in the mechanical forces. Okay, it allow the bone to respond to the change in the mechanical forces and facilitate the mineral homeostasis. Okay, so these are the three functions of the bone remodeling. Okay, okay, so now bone turnover rate. Okay, so in children we have 30 to 100% bone turnover rate. Okay, and in adult rate is quite slow. Okay, it is decreased in comparison with the with that of the children. After that, in the trabeculae bone, okay, in the trabeculae bone is more than that of the cortical bone. Okay, bone turnover rate. Okay, so here also I have drawn the flow chart. Okay, that I have taken from the Orbans, and again this flow chart is also very important. Okay, if a bone remodeling is asked in the exam, okay, so you can prepare this flowchart and you will get the proper marks. So in the first step we have quiescent phase. Okay, so this is a stage of the rest. Okay, after that there is a first phase. Okay, first phase is the activation phase in this bone remodeling. Okay, so activation phase, so first of all there is a detection of the signal in the form of mechanical strain or the hormonal action is there. Okay, okay, so mechanical strain is there or the hormonal action is there. Okay, so mechanical strain as I have written here. Okay, so mechanical forces respond to the changes in the mechanical forces. Okay, so that also causes the bone remodeling. Okay, so in the orthodontic treatments, okay, so the alignment of the tooths, okay, so alignment of the alveolar bone is changed. Okay, so that is due to the bone remodeling. So this activation state, there is a detection of the signal in the form of the mechanical strain or the hormonal action. Okay, and this causes the recruitment of the monocyte macrophages osteoclast precursor. Okay, so this just recruit the osteoclast precursor, and interaction of the osteoclast and the osteoblast precursor cells is there, and that causes finally the differentiation, migration and the fusion of the large multinucleated osteoclast. Okay, so this is quite written in the tough language. Okay, but you just remember activation phase means there is a activation of the osteoclast. Okay, activation means there is activation of anything, and that is osteoclast. So that is done by the mechanical strain or the hormonal action. So first of all you just recruit, and after the recruitation, okay, after the recruitment there is interaction between this osteoclast and osteoblast precursor cell, and that differentiate, migration also happens there, and fusion of the large multinucleated osteoclast takes place there. Okay, so this is the first phase. After that there is a resorption phase. Okay, so in the resorption phase, osteoclast dissolve mineral followed by the release of TGF, PDGF, IGF-1 and 2. Okay, and this cause the scalloped house shapes lacunae formed resorption tunnel or the cutting cone, and mononuclear cells macrophages remove the collagen remnants. Okay, so this resorption phase means there is a destruction of the bone. Okay, so there is a proper resorption. Okay, so in this point also there is a written same. Okay, so osteoclast it dissolves the mineral. Okay, and they releases the TGF, PDGF, IGF. Okay, and this causes the scalloped house shapes lacunae formation. Okay, and that is a resorption tunnel or the cutting cone. Okay, so this lacunae is a resorption tunnel or the cutting cone, and there we can see the mononuclear cell macrophages. Okay, remove the collagen remnants there. Okay, so there is a formation of the mononuclear cell. So don't go in detail here. Okay, just there you think resorption phase. Okay, and this bone remodeling is not bit more important, it is less important. Okay, it is a little bit less important than that of the bone formation. Okay, so just remember there is a resorption phase in the second step, and after in the last step, okay, after this resorption phase we have reversal phase. Okay, and the reversal phase, resorption cavities, okay, contain pre-osteoblast, apoptosis of the osteoclast. Okay, and new bone formation is there. Okay, that is new bone formation on the reversal line under influence of the released TGF, PDGF, IGF-1, IGF-2 in a resorption phase. Okay, okay, so in this reversal phase, okay, so there is a bone formation there. Okay, so resorption cavities contain the pre-osteoblast. Okay, so the portion where there is happening the resorption, it also contain the pre-osteoblast cells there, and after the apoptosis of osteoclast, after the resorption of the proper completion of the resorption of the bone, there is a formation of the new bone. Okay, so new bone is formed on the reversal line. Okay, and about this reversal line, about the resting line, we have discussed in the previous video. So I think you have concept of this reversal line. Okay, so reversal line is the line that is separating the resorpting structure, resorpting portion of the bone with the mineralization portion of the bone. Okay, properly formed structure, newly formed bone portion of the newly formed portion of the bone. Okay, so it is just separating that. So newly formed the bone, okay, new bone formation is taking place at the reversal line, and that is under the influence of the TGF, PDGF, IGF-1 and IGF-2 that was secreted here in the resorption phase. Okay, after that this new bone formation, so after the matrix formation there is a mineralization phase. Incorporation of the hydroxyapatite takes place there. This is by the calcification. We have discussed with this, okay, in a proper in detail. Okay, and finally again there is a question phase. Okay, so there is a cyclic pattern. Okay, so there is a, in this pattern there is a bone remodeling. Okay, so now there are some regulatory factors in the bone remodeling. So first is a systematic factor, another is a local factor. So in the systemic factor, okay, systematic factor, the hormone that decreases the bone resorption, okay, is the calcitonin and estrogen. Okay, so this table is also from the Orbans. You can learn this, and this is a those points can be framed into the MCQ question. Okay, after that increases the bone resorption. So high dose of parathormone hormone, okay, parathormone, okay, so high dose of PTH, parathormone, it causes the increase in the bone resorption. Similarly, high dose of vitamin D also causes increase in the bone resorption, and they are simple. I think you have learned about this in endocrine system. Okay, so calcitonin, estrogen decreases the bone resorption. High dose of PTH, high dose of vitamin D, it increases the bone resorption. Similarly, in the third point, increase in a bone formation is by the growth hormone, vitamin D, insulin, and low dose of the PTH. Okay, so those are facilitating in the bone formation. Okay, and decreasing in the bone formation is a glucocorticoid, and that is in a high dose. Okay, so you can remember this. After that, in a local factor, we have growth factors like IGF-1, 2, FGF, TGF beta, PDGF. Okay, so they are increasing the bone resorption. Similarly, the cytokines like interleukin 1, interleukin 6, okay, interleukin 11, PGE2. Okay, so they are also increasing the bone resorption. Okay, so also increase the bone resorption. So those growth factors are also regulatory factors for the bone remodeling. Okay, so they are not quite important. You just look all those points. Okay, okay, after that, markers of the bone turnover, bone formation, bone resorption. So alkaline phosphatase is also the best marker for the bone formation. Okay, bone turnover. After that, osteocalcin, procollagen first extension peptide. Okay, procollagen first extension peptide is also the marker. After that, in the, so they are the serum marker. Okay, so they are present in serum. Okay, so in the biochemistry, I think you have learned about those. Okay, after that, in bone resorption, we have urine calcium, urine hydroxyproline, okay, urine N-telopeptide, urine total pyridinoline. Okay, so those are the markers for the bone resorption, and for the formation we have those markers. Okay, they are present in the urine. Okay, so they are present in urine and they are present in the serum. Okay, okay, so I have completed this bone remodeling in fast. Okay, and this is not a much important topic, so I haven't spent much time in this. Okay, and video is also going to be bit larger. Okay, so I have discussed in a short, okay, about this bone remodeling. If you want to study more, then you can prefer the Orbans or prefer other books, okay, of the general basic science books, okay, of the biochemistry. Okay, so there you can learn in more detail about this bone remodeling. Okay, okay, so now we have completed all the topics for today's video. So now we will meet in the next video and the last video of the alveolar bone. Till then, stay happy, stay healthy, and study well. Okay, so today's video was quite frustrating, quite difficult to understand, but I tried my best to explain all those points clearly. Okay, so I think you have grabbed all those concepts, all those points. Okay, and if you can just take just your, if you can extract just 50-40% from this video, okay, then you will be doing good in your exams. Okay, so that is the main point to understand. So you don't have to be frustrated, but you just learn the things, okay, properly. So this in exam. Okay, if you just forgot all those point, okay, but you just remember this reversal phase, so that is also very important. So resorption phase, okay, reversal phase, mineralization phase, okay, activation phase. So those are the different phases of the bone remodeling. Okay, so telling those phases name, okay, also can get you the good marks in your exams. Okay, okay, so now we will meet in the next video. Till then, stay happy, stay healthy, and tata, bye bye, thank you.